Gel electrophoresis is a method that we use in our laboratories quite frequently to separate and analyze nucleic acids, including DNA and RNA. Agar itself is a compound that comes from cell walls of seaweed and red algae, and we utilize that compound in our laboratories in a more purified form called agarose. Now, when we make the actual agarose gels for electrophoresis, the higher percent agarose that you include in that gel, the pores themselves within that gel actually get smaller. Now, tonight, we're going to be utilizing here in my kitchen a compound that actually would do something comparable to agarose itself. And that is something that we have all in our kitchens everywhere, Jell-O gelatin. Now, gelatin is a processed collagen. It's a protein that literally makes up a third of your body, including your skin, bones, and tendons. This is a protein. So these, though, follow the same rules as agarose. The higher percent of gelatin, or Jell-O in this case, that we include in the recipes that we make, the smaller the pore size will be within that final material that you've produced, whether it be an agarose gel or actually whether it be Jell-O gelatin. So what I've done is I've heated up some water, and we're going to show you in this example that if we take a specific volume of water and increase the percent of gelatin in that specific gel, that would actually decrease, in theory, what the pore size would be by using just simply this household product. In the laboratory, we end up weighing out agarose weight per volume. So a 1% gel would be 1 gram of agarose in 100 milliliters of your buffer of choice. Here, all I want to demonstrate this evening is that the percent gel that you utilize as you increase it, that the same DNA molecules will behave differently in those different percent gels. And what we're going to use here tonight is grapes. So they're all basically going to be considered to be the same size of DNA molecule, and yet we're going to increase our percent gel. So let's get started. While the gelatin is cooling in the fridge, I want to give you a really simple demonstration as to the principles behind gel electrophoresis. So here I've got a couple of magnets, and one of these magnets is going to represent DNA itself. So because of the sugar phosphate backbone, it is negatively charged. So here we've got our magnet that's going to be DNA. Here we have a magnet that's going to represent the electrical field that will be behind the DNA itself, and where this DNA is actually going to run, we've got basically demoed as this example. Here, we'll consider this to be the pore, or a long pore that's running through the agarose gel, although the gel itself actually is made from thousands upon thousands of pores, and the pore size depends on the percentage of the gel itself. So we would turn on the gel, and the DNA itself would proceed to move down through the pores as the gel actually runs. And it would literally be repelled, just like the magnetic field between these two magnets repel them, because I have the two poles facing each other. Essentially, we could say it's a negative pole and a negative pole repelling. DNA and the electric field itself works the same way. The electric field will repel the DNA away and make it run towards the positive electrode. Alright, it's morning time. I'm going to be heading into work here in just a little bit, but we're going to get our gelatin out of the fridge, and we'll see what happened. So the first thing we're going to look at is the 1/4 times mix of gelatin. So the grapes, which represent the DNA, and the gelatin, which represents the agarose, the pore size is very big, and all of these same size DNA molecules can fit through there very, very easily. Let's look at a higher concentration. Here, this was 0.5x, 1/2 pack. And you can see the gelatin's moving, kind of funny, but it's not nearly as rapid than the 0.25x, than the 1/4. Here is the 1 times the 1 package of gelatin. You can see this is just like normal gelatin. This is what you'd expect to see. The pore size is very, very small now. Here is 2 times. 2 bags. 2x concentration. So there you have it, a demo on how pore size can be affected by the percent of gelatin or by agarose that's present in agarose gels when we do gel electrophoresis. Just like the right concentration.
See, when you get all the kids together and you say, "Kid, you want a treat?" They say, "Yeah, we want a treat." So we get the Jell-O all out of the trees and say, "They got big Jell-O, blue Jell-O, green Jell-O, red Jell-O. They got all types of Jell-O. We can cut them in squares, dirt, stops, and come on, J-E-L-O."
Okay.